Good afternoon, everyone, and thank you for joining us for today's webinar. My name is Josh Vickers, and I am with RTI International's Forensic Technology Center of Excellence, and I will be your host and moderator for today's event. Today's webinar is entitled Effects of DNA Extraction Methods on Recovery, Degradation, and Loss. We do have three presenters and subject matter experts for today. Our first presenter is Dr. Brian Kemp. He is an associate professor in the Department of Anthropology and co-director of the Laboratories of Molecular Anthropology and Microbiome Research at the University of Oklahoma. He has 20 years of experience studying DNA recovered from aged, degraded, and low copy number DNA samples. He has published 68 papers that address numerous topics, many of which on the evaluation and improvement of methods for recovering DNA from age, degraded, and LCN sources. Our second presenter and subject matter of the day is Dr. Kara Monroe. She is a research associate in the Department of Anthropology as well, and at the Laboratories Molecular Anthropology and Microbiome Research at the University of Oklahoma, with specialties in paleogenomics and K-12 STEM outreach. Dr. Monroe is currently the lead PI on a multi-year project funded by the National Institute of Justice that focuses on next-generation sequencing and degradation and retrieval of touch DNA. She's also a co-PI or lead researcher on 3 additional NIJ-funded grants funded by the same institution. She has over 17 years of experience studying DNA recovered from aged, degraded, and low copy numbered DNA samples. Our third presenter of the day is Dr. Christine Beatty. She is a postdoctoral research associate at the Laboratories of Molecular Anthropology and Microbiome Research at the University of Oklahoma. As a graduate student of the University of Kansas, she utilized modern and ancient DNA methods to answer anthropological questions of human migration, admixture, and identity, focusing on people in the Americas, in addition to managing the laboratory and training students. She currently works with her postdoctoral advisor, our first presenter, Dr. Brian Kemp, to better understand how standard laboratory methods can alter, damage, and lead to the loss of DNA through the use of NGS sequencing, fragment analysis, and bioinformatics. Just our quick disclaimer, the opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenters and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. And now, without any further ado, I would like to hand the microphone and platform over to our first presenter, Dr. Brian Kemp. Dr. Kemp?
Okay. Oops. All right, okay. Thanks for that nice introduction, Josh. I'm very excited today to have some of my team members and I discuss some of the results that we've gotten from our current National Institute of Justice project, which is a 3-year project. We're about a little over halfway through the project. You'll note that we've added a co-presenter here, Dr. Krithi Sankar. He is also a co-PI on the grant and contributed to some of the analyses that Dr. Monroe will talk about later. The way that we're going to structure this is that I'm going to introduce the theoretical background, like what questions drive our research and why, and then Dr. Monroe will take over and talk about a set of experiments in which we evaluate the efficacy of various DNA extraction methods on the retention of DNA standards. And then lastly, Dr. Beatty will discuss another set of experiments that aims to track down the nature of the loss of the DNA that we observed during DNA extraction. So the question that my research team and I have been thinking about for a while is this, and it's a simple one. It's how much DNA is obtainable from any given source. And you might think, well, you can open up a manual, a DNA extraction manual, and it'll say you get a certain number of nanograms from liver and a certain number of nanograms from hair or any kind of tissue. But what we've come to quickly realize is that those estimations of the amount of DNA that you get from various tissues is only the DNA that you retain through the process of extraction and purification. So ultimately, the amount of DNA that's available from any given source can be broken down into this formula. So we get a certain amount of DNA, and that is a product of the original amount that was present in the tissue, which is unknown, which is then compounded by, say, loss during sampling. And in this case, I'm really thinking more about in the case that, say, you're trying to swab DNA from a location. So how much do you lose during that sampling? So that will impact the amount of DNA that you retain through the process. Then the amount of DNA that's associated with the extraction and purification of that DNA. And lastly, loss in quotes here due to amplification bias. And what I'm really talking about here is the role of PCR inhibitors, the presence of which can skew your vision or your estimations of how much DNA is actually in a sample. Now, the key here is that each one of these variables is typically unknown, or at least is poorly characterized. So the question of how much DNA is available in any original tissue or any original source is unknown. We're going to focus here on trying to estimate how much DNA we lose during the extraction and purification process. And that forms the basis for the rest of the talk. Now you might be thinking, well, I've read plenty of papers where they compare some number of DNA extraction methods. In this fictitious example, let's say we're comparing extraction method A, B, and C against some tissue here, a bone, a rib in this case. Okay, and we can then extract a piece of DNA, or we can extract DNA from a piece of rib using each method, and we might determine that extraction B is superior in this case to extraction A and C. Well, that's great information, but we have to realize that this comparison is a relative one. It does not tell us the absolute efficiency of extraction method B. It only tells us it's better than A and C. It could perform very, very poorly, but in this case, we don't know. And the vast majority of comparative studies, or studies that have compared extraction methods, have used exactly this approach. So their best method is the efficiency relative to 100% is unknown. Okay, that's important to keep in mind. There have been a few studies, I'm going to highlight them here, that have actually used DNA standards. So a known amount of DNA entering the system is then extracted and then compared to the outcome. So in that case, the outcome, the amount that is retained through the process, can be directly compared to the input. And this will give a precise estimation of the efficiency of the extraction method, an absolute efficiency, not relative. The first study that we know of was published in 2010 by Lee et al. And what they did was they took DNA standards of 25 nanograms. Some of the standards they kept as intact genomic DNA, and other standards they treated with a DNase to break down the DNA to some degree to make it represent degraded DNA. Okay, so both standards were 25 nanograms. One was intact, one was degraded with DNase. Then they tested a variety of QIAGEN-based extraction methods, and in some cases they changed the components from one kit to the other, but it was all based on silica-based extraction and QIAGEN products. And the outcome, they used to quantify the outcome, they used the quantifier human DNA quantification kit. And what they found was that the best method of the various QIAGEN methods that they used retained 38.9% of the intact standard. Interestingly, it retained the best method that for the degraded standard retained 50.8%. So in this case, they were able to retain more of a degraded standard than an intact standard, which is an interesting observation. But you still note that this means that the DNA loss here is 50 to 60%. One drawback to this study was that they didn't characterize the degree of degradation that was caused by the DNA's treatment. So what they could have done is took the DNA's treated DNA and run it on a gel just to get an idea of the size distribution of what they were working with, but they didn't do that. So we don't know how degraded the DNA was, despite the fact that more was retained. And that would have been an interesting thing to quantify, but they just didn't do it. The second study that we know of that used a standard methodology, as I've been discussing, was this one by Dabney et al. In 2013. What they did was they took a standard mixture of 5 no-limit DNAs. These are size standards of 35, 50, 75, 100, and 150 base pairs, and they combined those into a mixture at 285 nanograms. And then what they did was they extracted that mixture of DNA with a method that's called the Rowland et al. 2007 method, which is a very popular method used by ancient DNA researchers. And then they did slight modifications to the Rowland et al. Method, and they compared that as well. They compared their DNA in to DNA out with a bioanalyzer, which quantifies and qualifies the DNA standard, so they could see what was happening to the quantity and the quality of the DNA in and out. And what they found was this. Here we can see the Rowland retention in the light gray, and you could see that as smaller fragments were associated with greater loss, larger fragments with greater retention, and so forth. But some, the loss ranged from somewhere from 80% to 20%. Their optimized method, or the modified version of the Rowland method, you can see improved quite a bit the retention of all base pair size fragments. And what's interesting here is that the, let's say 95, I don't know the number exactly here, but let's say like about a 95% retention of a 35 base pair fragment was interesting because both of these methods utilize the minolute column, the QIAGEN minolute column, which is supposed to retain DNA fragments of 70 base pairs or higher. So it should get rid of base pairs less than 70, but here they're getting 95% retention of 35 base pair fragments. So that's a pretty interesting observation as well. The cause of the effect of the improved retention of the optimized method is impossible to determine because they simultaneously changed multiple variables. It's great that it works really well, but it would be interesting to know if it was one particular variable that led to this increased retention of these small pieces of DNA. But we don't know that because they didn't change one variable at a time. But still a very interesting study. And my research team played a role in this as well in publishing in the Journal of Archaeological Science in 2014. And what we did was we created standards using amplicons of 181 base pair fragment of a northern fur seal. So we took northern fur seal DNA, we amped up 181 base pair fragment, we purified those fragments, and then we could dilute, we used qPCR to then be able to dilute those standards down to any number of copies. And in our paper, we were discussing copy number, not concentration, but basically in this study, we looked at copy number variance ranging from 13,000 copies of that 181 base pair fragment up to 50,000 copies. And if you calculate that, that's down in the femtogram level. So it's a very low amount of DNA. And then what we did was we used qPCR to get an estimate of DNA in, and then we would extract that DNA using a variety of different methods. For example, phenochloroform, we looked at the influence of isopropanol precipitation, various silica-based methods, and some kits here, okay? And what we found was that across those kits, we would lose somewhere between 45 and 99% of the 181 base pair fragment, which was pretty astounding to us at the time. This was not what I expected at all. In the ancient DNA literature, one of the ways that ancient DNA researchers authenticate their results is that they make the claim that ancient DNA must be in low copy number. The expectation is if you have too much DNA, it can't be ancient DNA. What we argued in this paper here is that we might actually be helping create the condition of low copy number through the DNA extraction method, and then we are causing the DNA to take on the characteristic that we then use to authenticate the results. This would be highly problematic, as you can imagine. One limitation to our study is that we looked at a single-sized piece of DNA, 181 base pair fragment. So in this case, we don't know how other sized fragments would behave under the same conditions. And another thing that occurred to us is that some of this DNA loss might actually be caused by fragmentation of the standard or shortening of the DNA strands. In the case that the DNA becomes fragmented and loses priming regions, you wouldn't be able to count it during qPCR. So some of this loss, although we could not tell in this experiment, some of this loss might actually be due to the inability to prime and amplify the strands because they become shortened. So that's why I put loss in quotes. We can't assess if that is the source of loss, but it might be. And some of what Dr. Monroe will talk about will address that. So then also in 2014, we published a paper in Human Biology to improve upon our look at DNA loss of various sized fragments. And what we did was we took the PowerPlex 16 kit, which probably some of you are familiar with. And what we did was they published their primers, so we ordered the primers individually. So we didn't use it from the kit. We ordered the primers individually and then amplified from human DNA all 16 pairs of primers, creating 16 different fragments ranging on the low end from 106 base pairs and on the high end to 428 base pairs. And then again, what we were able to do is take those 16 fragments, we were able to pool them and count with qPCR how many copies we have. And then we were able to dilute them to copy number standards that we wanted to evaluate. In this case, we looked at copy number standards ranging from 10,000 per microliter to 10 million per microliter. And if you calculate that in concentration, that's from the femtogram to nanogram standards, but pretty low, as you can see. And in this case, we wanted to test the effectiveness of the QIAGEN minilute purification kit in the retention of these molecules. So we put molecules through that system, how many molecules come out. The minilute purification kit has been used increasingly in ancient DNA endeavors. As I mentioned, the Dabney study used it. This is because this column is said to be able to retain 70 base pairs or higher, and that's a quality that people appreciate about the kit. I've also seen that this kit is becoming increasingly used in forensic investigations as well. Across all of our standards, we got the average loss of about 40%, so retention of about 60%. What I'm showing you here are 3 different standards at 3 different orders of magnitude, 10 to the 4th, 10 to the 5th, and 10 to the 7th. And then the average loss here regressed against the fragment size here. None of these lines are significant, so there's no difference in the retention using the QIAGEN minilute purification kit of base pairs of around 106 up to 428. So we lose about the same amount of every size fragment. That was unexpected. A drawback here is still we're using an amplicon-based approach. We could certainly improve upon that by using genomic DNA, which is one of the I think, the improvements that we made in our current National Institute of Justice endeavors. The experimental design for the part that Dr. Monroe will discuss, and it's very similar to what Dr. Beatty will talk about later, is that we created standards here from full genomic DNA. In this case, we acquired a full pig liver. We did extractions from that liver. And one of our standards we made at 100 nanograms per microliter at about 7,000 to 9,000 base pairs in length. So this is what we would call fully intact genomic DNA. Then we took that standard and diluted it down to 1 nanogram per microliter, but very intact, still 7,000 to 9,000 base pairs in length. And we wanted this to represent maybe what we could expect from fresh touch DNA, where the amount of DNA is low, but it can be very intact, especially if the touch DNA is fresh. Third, we took that standard 1 and we sonicated it down to 250 base pairs in length. So we have a highly concentrated, highly degraded sample, and this is what we call an experimental state. We're not sure that this is something to be expected to exist in the natural world, but we thought it was an interesting data point or an interesting standard to compare. And I'll show you why, because it fills in an interesting space in what I call parameter space. And then 4, we took that third standard and diluted it down to 1 nanogram per microliter. And again, it's degraded to about 250 base pairs in length. And here, what we wanted this to represent is lower copy number DNA, something that's degraded genomic DNA, okay? Now, what I mean about parameter space is something like this. Our standard 1 is long strands and high concentration. Our standard 2 is long strands but low concentration. Our standard 3 is short strands but high concentration. That's the experimental one. And then the last one is both degraded and in low copy number. And so we wanted to look at the behavior of these different standards as we put them through various extraction methods. We have a couple expectations that we thought of. Any given extraction method could result in one of these 4 outcomes. The first one, labeled A here, what we're seeing here is this would be concentration, RFU, and over here would be base pairs where the length of the DNA is larger here and shorter here. We could imagine that a DNA standard going into a system could simply lose concentration and shift to the, or sorry, be degraded with regards to strand length, where we see the concentration doesn't change, but the average base pair has been decreased through fragmentation. B, another possibility is that the DNA standard in is both degraded with regards to strand length, and we lose DNA through the process here. So degraded and lower concentration. Under scenario C, we can imagine a DNA standard going in where you simply lose DNA and decrease the concentration, but the average base pair stays the same. There's no fragmentation. And a fourth possibility is that the DNA in here, we preferentially lose smaller pieces of DNA, shifting the average base pair to a higher average, but the concentration is less. Those are our 4 expectations. And we wanted to see how that applies to these various extraction methods that we tested. Our goals were pretty simple for this first part. We want to document the influence of various DNA extraction and purification methods on the quality and quantity of resulting DNA elutions. How much DNA goes in? What is its quality? How much DNA comes out? What is its quality? We are not here trying to determine a best method. That was not a goal. Even if we determine that something performs well, this was not our goal. Now I'm going to shift over to Dr. Monroe. She'll be taking over.
Dr. Monroe, it looks like your microphone might still be muted.
Thank you.
There you go.
Thanks, Dr. Kemp. Briefly, I wanted to go over our basic experimental design and the methodologies, and then that'll be followed up by some preliminary results looking at both recovery of total concentration of DNA, and then followed by looking at fragmentation rates across different extraction purification kits. So briefly, we had 20 different extraction methods. Some of these were different methods from one type of extraction kit where they might have different kinds of buffers, and some might be just completely different kits across different manufacturers, as well as some kind of homemade kits for ancient DNA or paleogenomics, where they're taking columns from one kit and maybe buffers from another that are commonly used in that field. So just as Brian had, or Dr. Kemp had described, we have standards 1 through 4. And so for each of those standards 1 through 4, we have 10 replicates. So standard 1 will have 10 replicates, standard 2 would have 10, et cetera, et cetera. Standards 3 and 4 were degraded, and they were sonicated using a Q-sonica to approximately 200 to 250 base pairs. Okay, I'm having some issues moving forward here. Just a second. Okay, so all standards were quantified using the qubit 3.0 fluorometer, both pre and post extraction. And then each of those 10 replicates for each standard was also quantified with a qubit. The sample replicates also were then run on the fragment analyzer 3 times to determine average fragment lengths as well as concentrations. And so this allowed a good comparison between concentration values that you're going to get from the qubit and then comparing that to what we're getting from the fragment analyzer. And both use a fluorometric kind of measuring system, but then with the fragment analyzer, you have a gel electrophoresis kind of baseline rather than simply just having a solution with like fluorometer going through it. Okay, so with the 20 different kits and different methods, we divided it up into 3 different kind of uses. So ones that were targeting both forensic and touch DNA or situations, followed by ancient DNA and paleogenomics, and then kind of standard kits that people use for just general tissue extraction or any kind of common laboratory extraction that you have. We also wanted to touch base with having kind of a across different types of extraction methods. So in this case, with the 7 different forensic touch kits that we're looking at, some are based on a silica kind of membrane or a column, and then others are focusing on, like the prep violet kit, focusing on a bead extraction method. With the ancient DNA, we looked at 5 different kits, but with variations within each of those extraction methods. The first would be, as Dr. Kemp had already talked about, is Dabney et al., which is using a modified minilute protocol with different centrifugation speeds, but also using much larger amounts of lysis buffer. So rather than, say, the minilute is using approximately 700 microliters, they're using up to like 12 to 14 mils. So definitely flushing through more solution through those columns. The second ancient paleogenomic method we're looking at has 4 different types of methods in there. 2 are using Roche viral spin columns that are also silica based. And then due to the cost prohibition of those columns, they also offered alternatives using 2 different buffers, but with beads rather than the silica column. We also looked at Kemp et al. 2007. That is basically a 3-part extraction method that is phenylchloroform, followed by alcohol precipitation, and then a silica slurry extraction as the final stage. And then lastly, we have Kemp et al. 2014. That is a modified version of 2007, which excludes the phenylchloroform and also excludes the isopropyl precipitation, and then varies the amounts of buffers that go into the silica slurry. And lastly, for the ancient DNA kits, we have Mobile or MP Biomedical's Gene Clean for Ancient DNA, which also has 2 variations of different buffers that you could use. One targeting more for inhibition versus not. I also wanted to go back and add the Rowland et al. Was specifically trying to retrieve extremely small fragment sizes, so approximately 35 base pairs. And then lastly, with the modern DNA kits, we have Bio-Rad's InstaGene Matrix, which is a Helix-based extraction method, and then the Minaloop PCR purification kit, Kaya-AMP, and DNA blood and tissue handbook, or blood and tissue kit, which are all silica column-based extractions. So moving into the quantity and documenting fragmentation size, we started out wanting to compare qubit and fragment analyzer and differences and similarities that we might see in how to quantify both our standards as well as our output after going through the different extraction methods. And both have some pros and cons to what they can offer you. So the fragment analyzer is similar to the tape station, also by Agilent, which provides both fragment and concentration. So it gives you a concentration as well as fragment sizes. It also allows you to look at inclusion and exclusion of different fragment sizes. So if I wanted to know total concentration of, say, 100 base pairs to 500 base pairs, I could do that and exclude all else outside that range that I wanted to look for. It's also high throughput and high sensitivity to compare to the qubit. So we're looking at, 0.005 nanograms per microliter compared to, say, 10 picograms for a qubit. So it has much higher sensitivity. On the downside, it is, and from what we found with our experimentation, it's very sensitive to overloading. And so as a result, you really, to have effective concentration sizes and consistency across runs, you really have to have an initial quantification with something like a qubit or a nanodrop, and then have dilutions to fit accordingly. So the machine ultimately works better with smaller concentrations versus larger. And so what tends to happen with the overloading is an example shown here. And there also seems to be an inverse relationship with a fragment analyzer between size accuracy and concentration accuracy. So the top image demonstrates undiluted standard going into the fragment analyzer, and the output then shows almost a complete accuracy to what the input concentration should be based on the qubit results. Unfortunately, what you then see is with the increased accuracy of concentration is a shift down in the fragment size, which becomes much more inaccurate. So in this case, a diluted sample of the same standard would have approximately 7,000 base pairs here as far as its fragment size versus what it's showing here undiluted at 1,700 base pairs. So what you do have is the shift with fragment size and concentration, right? They have this relationship, and the key to having consistent results is to find that sweet spot in between the two options. It's also sensitive to overloading your ladder or underloading your calibration ladder, which again will shift your concentration sizes as well as your fragment sizes. So as a result, we have this high run-to-run variance. So the general genomic kit would say it would have on average plus or minus 25% run variance. Some of this has been mitigated since Agilent has taken over manufacturing both the kits and the machine versus advanced analytic. And there's a link provided here that shows, rather than manually pipetting, actually shaking your samples for 5 to 15 minutes can drastically reduce the inaccuracy or run-to-run variance that you might see. Okay, so just briefly, I want to look at standard 1 and standard 4. So standard 1 is the intact genomic DNA, and just comparing the concentration values that we're getting, as well as the standard deviation that we're getting with the fragment analyzer versus the qubit. And so we can see there's some drastic differences. In some cases, we have the qubit value with showing absolute amounts of DNA with the fragment analyzer showing almost none, but we're also seeing quite high standard deviations with the fragment analyzer results. We can also see just with this, and then although this might all be affected by the extraction itself, standard 1 is also showing a complete deviation from what the qubit value shows of what we thought our input would be. So we're coming out with 200 nanograms when the qubit in our dilution should have been 100 nanograms per microliter. And the same thing we can see with standard 4, which is low copy and degraded. So again, at 1 nanogram per microliter at approximately 200 to 250 base pairs. We can see some drastically different results between different kits. This would be an example of instagene. So the qubit value is giving much higher values compared to the fragment analyzer. Same with the DABNY protocol, much higher qubit values with no fragment distribution or concentrations of DNA on the fragment analyzer. And we can also see the reverse, where we might have higher, in this case, the investigator kit from QIAGEN, having higher amounts on the fragment analyzer, but less on the qubit. So this is causing the conundrum of trying to figure out why is there this inconsistency between the qubit versus the fragment analyzer, despite the 25+% variation that you have. It's still not being accounted for. Where is the DNA going from the qubit values from the fragment analyzer values? And why is that, what is going on there? And, you know, qubit can be inhibited by salt, phenol carryover, alcohol carryover. The same with the fragment analyzer. High salt could also inhibit the results you're getting. Although, as we stand, we're still trying to figure out the exact relationship of why different quantification methods are giving you different results. Moving forward, just to look at the total impact of extraction kits and total DNA recovery, we're going to be using values from the qubit. And then later, when we talk about fragment size recovery, we're going to use data from the fragment analyzer. Okay, so looking at standard 1, look is again intact genomic DNA, 100 nanograms, approximately 700 to 7,000 to 9,000 base pairs. We have the codes for the different kits that you'll see here, and then also note that we have color coded the methods. So ancient DNA methods are coded in blue, modern DNA extraction kits are in yellow, and then touch forensic DNA kits are in gray. We also have kind of just a quick breakdown of the table where we can see greater than 75% recovery of standard 1 was only the Rowland using a spin column with buffer G. The rest are kind of breaking down between 25 and 49% equally with 2 ancient methods, 2 modern methods, 2 touch methods, and 50 to 74%, again, 2 ancient methods, 2 modern, and 1 touch. And we've highlighted in the kind of dashed boxes here, the ones that are 50% or above. Also note that some data here, Kemp 2007 method, had a recovery of over 50%, but only had 2 data points, thus saying the other 8 data points had no DNA. So in this case, while you had high recovery for some, it was highly variable recovery rates. Standard 2, which is kind of meant to mimic touch DNA, which is at 1 nanogram per microliter, 7,000 to 9,000 base pairs. Again, we're kind of seeing a shift where we again have the Rowland using viral spin columns with the buffer G is performing very well at over 75%. But we also have additional trace nucleotide spin trace kit improving its methodology. Standard 3, which is the experimental state, 100 nanograms, but sonicated to 200 and 250 base pairs. We can also see improvement in overall general improvement of kits with this experimental design. So kind of showing that in this case, lower concentrations, or sorry, lower fragment size might improve yield with some of these different methods. Again, but we're seeing Rowland's method using columns, but in this case, a different buffer is improving the yield. Looking at standard 4, low copy number degraded genomic DNA, again, sonicated to a small base pair length at 1 nanograms. We can see, again, the Rowland column methods still performing at above 50%, but slightly reduced from before. And also some of the kits having more higher variance with standard 4 versus not. And then again, some improvements over other standards. Okay, so quickly shifting gears, we're going to look at the impact of extraction kits on DNA fragment distribution. I'm going to briefly go through a root mean square deviation analysis that's based on the fragment analyzer data. And again, just kind of a refresher, the samples were run on the fragment analyzer 3 times for each standard. So we have 10 standards, or 10 replicates for each standard, and those were run 3 times on the fragment analyzer. And then looking at various size ranges, looking at the concentration across different size ranges. So for this analysis, we had 4 fragment input ranges, and hoping to like mimic what we would see within a laboratory setting. So between 20 and 100 base pairs, we had classified as low copy number degraded DNA, and then 100 to 500 base pairs is kind of mimicking what you'd normally see with CODIS size ranges or next generation library size ranges. And then 500 base pairs to 10,000 base pairs would be the mid-sized genomic range. And then 10,000 to 30K BP would be your high molecular weight, which normally you wouldn't see unless you're doing single cell sequencing or something with PacBio. Okay, so with standard 1, looking at intact genomic DNA, what we're trying to do here with this root mean square deviation is it's kind of a 3D geometric way of looking at a list of paired points across space and measuring that distance between those points. So our class ranges are those points. And we want to see how samples are deviating from those points. So basically, we're taking the output concentration within those size ranges minus that input concentration for each of those 4 size distributions. We square that, and then we take the square root of the sum across all forward squared values. So in this case, what we're seeing within this chart is the lower the percent deviation, the less that kit is impacted by variation in fragment sizes across all 4 distribution. So in this case, we would have QIAGEN Minilute kind of hovering around 10% deviation, which means it's only deviating across all of those 4 space classes or size classes by 10%. So in this case, it's being less affected in fragment size than, say, something that's higher at like 15%. So to give a better clarification of what is happening with potential fragmentation would be an example. In this case, we're going to look at one that was particularly high, the Thermo Fisher Prep Filer kit. And so what we can see when we actually look at the fragment analyzer images of the fragmentation rate. So we can see is here's our pooled standard. So that's kind of our control that we run with each set of replicates. And then these are just some examples of the DNA fragmentation that we see post filer extraction. And so what we can see is kind of what Dr. Kemp was talking about earlier, where in this case, you're losing concentration, but more so you're losing, you're getting high fragmentation, almost in orders of magnitude. So you're going from 7,000 down into like the 300, 400 base pair range. Other examples would be the CIAMP DNA Investigator Kit, again with standard 1. And what we can see here is actually hardly any fragmentation at all. Maybe some slight loss of concentration in some of the replicates, but in general, you're maintaining concentration as well as fragment size. So less fragmentation rates going on. And interestingly enough, in this case, the fragmentation we do see is some degradation of our standard. Okay, looking at standard 2, which is supposed to imitate what we'd see with potential touch DNA. Again, we're kind of seeing variability in fragmentation rates, both increasing or decreasing depending on what standard that you're looking at. There appears so far to be no standard pattern of fragmentation from one standard to another. Although in this case, overall things seem to be increasing with that lower concentration size. So an example to give for standard 2, looking at nucleus spin DNA trace. In this case, nucleus spin trace was showing a larger increase in fragmentation for the standard. This is why it's probably especially important to actually look at your data and see what kind of anomalies may be appearing. So in this case, what we can see is not less fragmentation, but some blips that are increasing in fragmentation size. And so this might be just noise. It could be small amounts that just by chance are getting picked up for these replicates, when maybe across some of the other replicates you're not seeing it. And so this is maybe artificially skewing the deviation rate for this particular kit. Some other examples that we might see with the experimental state N3 would be some increases in, say, nucleus spin trace kit, where you have an increase in the deviation. And what we'll see there is an example of what Dr. Kemp was talking about, where you have your sample in and out, in this case, around 300 base pairs. This is your standard compared to the nucleus spin trace results. And what we can see then is probably what is happening is we're preferentially losing small fragment sizes, decreasing the overall concentration, and then less obscuring the smaller amounts of higher fragment sizes that might remain in this distribution. So in this case with our standard, there might still be small amounts even after being sonicated that are around 2,000 base pairs, but it's being obscured by the higher concentration at its height peak, which is around 200 to 300. In this case, since we're losing so much more of the smaller sizes, it's actually picking up some higher fragmentation range. And again, in this case, it might be more artificial deviation in fragment size rather than truly higher distributions of fragment sizes across. If anything, you're probably losing more smaller fragment sizes. With standard 4, low copy number, degraded genomic DNA, again, we're kind of seeing some increases and decreases depending on the kit as far as their fragmentation rate across the different standards. Next, I'm going to highlight the prep filer. And in some cases, what we'll see is in this case, the gray is the pooled standard. And what you'll see then is the fragmentation rate is increasing because you're losing your DNA entirely. And so in this case, the concentration sizes are so low and spread across off so many, or actually a total loss of DNA, that's actually showing a higher deviation in fragment size due to total loss rather than actual fragmentation within what's able to be picked up. So DNA and DNA. Okay, so this is just hard to kind of see all the different charts and see the patterns. So this is one image, just kind of pulling it all together and then divided it up by the different kinds of kit methods that we have. So this is looking at fragmentation rates across all of the touch DNA, fragmentation rates across modern DNA, and fragmentation across the ancient DNA kits, and then with the different standards. So the blue is standard 1, green is standard 2, light blue is standard 3, and red is standard 4. And so what you can see is that different standards are behaving very differently for the different kits, regardless of type of kit they are. Although there is some more consistency with the QIAGEN Minilute and QIAGEN Micro. Okay, so some basic conclusions. I would argue that we're at a point where there is so many different ways of measuring concentration, and none of them are fully effective for what you really need. And so what you have is a lot of maybe potential inconsistency of your qubit versus your fragment analyzer. You might see the same thing if you're using a bioanalyzer versus a nanodrop. And so when we're doing these experimentations, we have to keep in mind looking at all these different variables and trying to figure out what is potentially the true concentration, or at the very least looking at how they relate to each other, and especially how this might relate to fragment size. Generally speaking, the extraction methods vary widely, both in recovery and fragmentation across all standards. So the standards are a significant variable, but so is the extraction methods. So it's hard to tease out which one has more power in predicting what the outcomes are going to be. But it does seem clear that some kits perform better than some kits do perform better with certain size ranges and concentrations than others. Ancient DNA methods optimized for small fragment sizes. Overall, what was interesting is that they did perform well with other standards, even though the goal was to get ultra small fragment sizes. And also I want to clarify as well that we cannot account for real world variables such as inhibitors or bound DNA matrices, things like that. And I say that because some of the kits that did not perform well in this experiment, I have used for paleogenomics or I've used for ancient DNA and have gotten great results. And so there are other mitigating factors that need to be looked at after these experiments to kind of further understand what's going on with endogenous DNA that is compromised. Okay, so I'm going to pass along to Dr. Beatty.
Okay. All right. So Dr. Kim and Dr. Monroe have talked a lot about experimental design and talking about the potential for DNA loss with these DNA extraction methods. And after going through all of these processes, we realize, you know, one, we're seeing a lot of DNA loss, but where is the DNA going? Uh-oh, my thing does not want to move down. Okay. So what we know is that the behavior of DNA during a typical DNA extraction or purification is poorly understood, right? And we know that DNA is lost during these practices, but we're not sure where, and we're not sure what the DNA that we retain looks like necessarily. DNA loss can occur in multiple ways, right? The cells might not lyse, DNA might bind to plastic consumables, but what we know we can focus on here in this project is that we can see whether or not all the DNA binds to a silica, And we can see if DNA does not efficiently wash off silica. So we're going to focus on a single extraction method here and take a look at what happens if we modify it for this experiment. So we have a pretty simple goal. We're trying to determine where DNA loss occurs in a commonly used DNA extraction kit, with the hope that we could take this information and direct protocols and modification to increase DNA yield. So again, we start off with 4 standards. These are not from the original study. These are standards. We ordered genomic DNA from Ziogen. These have a larger sort of peak fragment length of about 20,000 base pairs. So we have concentrations of around 100 to 120 nanogram per microliter of intact DNA as our standard 1. Standard 2 is, again, intact DNA at a lower concentration of about 0.6 to 0.8 nanograms per microliter. Standard 3Z is our fragmented standard, where most of the fragments are between 250 and 350 base pairs in length. And again, this is an experimental standard at a high concentration. And standard 4 is also a degraded standard, but at a lower concentration to represent something we would see in a degraded sample. Here we're looking at QIAGEN D and EZ, and we didn't select this kit necessarily because we thought it performed better than others. It's just a kit that we tend to use most often in this lab when we're dealing with a modern, what we would expect to be an intact DNA source. So it's a guanidine-based extraction method that utilizes a silica spin column. And it actually had more information in its manual than most kits do as far as what you can expect to receive as an output from DNA. So it tells you that you can get fragments up to 50,000 base pairs in length, but most of your DNA should be around 30,000 base pairs in length. And the yields are going to vary for blood. It says that your net yield should be around 2,000 to 6,000 nanograms, but you get up to 40 micrograms from tissues such as a rat tail or bird blood. And since we are using a pure DNA, we weren't really sure going in where our samples would fall on this range. So I'm going to walk you through the basic experimental design. We basically start with a normal DNA extraction, just like most of you have done probably hundreds of times. So we take a lyse standard and we put it through a silica spin column so that it can bind. Once the DNA is bound to the silica column, we move it into a new column, a new collection tube. And then we'll try to remove the DNA from the collection tube by putting in an elution buffer. So now what you see on the screen here, I don't know if you can see my mouse or not, but we have a tube that has a flow through. So whatever DNA did not bind to the silica column is left here. We have all the DNA that was washed from the silica column here. And now we're going to move the silica column over into a new collection tube here. Okay. And again, we're going to add a elution buffer and remove whatever DNA is found on this second column. We're going to move the silica column again into a third column. And we're going to keep doing this so that we can wash whatever DNA that is bound to the silica column that we didn't remove from it in the previous attempts. So now we went back to the flow through from our first sort of extraction run, and we're going to put this through a new silica column. We're going to have it so that it binds, oops, I went a little too fast, so that it binds, the DNA has a chance to bind to this new silica column. And then we're going to wash away the DNA with elution buffer, and again move that silica column up through 10 tubes so that we can see how much DNA is still retained in any of these sets here. So this looks like a lot. So basically what we did is we took one standard, the standards of high concentration really for this large size that we see here. We're going to put that one standard, we're going to put 200 microliters of it through a normal DNA extraction through one silica column, and then we're going to keep keep adding elution buffer to that silica column in separate tubes to see if we can detect any DNA in those other tubes. And we're going to keep on doing this with the flow through, moving flow through into a new silica column over and over again until what we end up with for the high concentration standards are 100 tubes that we can use to detect if there's DNA present, if it was stuck on the silica spin column, or if it was trapped if it didn't bind efficiently in the first place and ended up in the flow through. For the low concentration standards, we scaled this down a bit so that we ended up putting the standard through 5 spin columns and putting the flow through those so that we end up with a total of 25, I'm going to call them samples, but they're basically 25 outputs from this original standard. So we took all of these and we quantified them using a qubit 3.0 fluorometer. And again, we ran these samples on an Agilent fragment analyzer to determine both the average fragment lengths that we see in the sample, but also with some of the lower quantity concentrations, which we couldn't detect on the qubit to see whether or not DNA was present in those. So here we're going to see some examples where the fragment analyzer is more sensitive, so we actually can see that there's probably DNA there where we couldn't see it using the qubit. So first I'm going to show you a few of the results from standard 1. So standard 1 is our intact 20,000 base pair fragments is the sort of the average for this standard, and the qubit value was about 118 nanograms per microliter. So this is the standard in here, and this is all of the products from the first silica spin column being moved from the first attempt at an extraction and then moving the silica column through into new tubes. So this first column, we retained about a sample that had 31.4 nanograms per microliter, which is actually fits in pretty well with what DNEZ says you can get out of a silica spin column. And then we notice that we still see detectable amounts of DNA from the qubit all the way through all 10 of these samples, up to where there's even around 2 nanograms per microliters of DNA in this final tube here. And that's actually enough DNA to do a lot of downstream analyses. So that's quite a bit of DNA. What you're looking at here are results from the fragment analyzer. So what you have is here is the standard in, and each of these colors represent different fragments of DNA. So the lower blue down here is 20 base pairs to 100 base pair fragments of DNA. Our average, most of our DNA in the standard is between 10,000 and 30,000 base pairs in length. So that's this big bar here. So what you see is over time, at first we start to recover these larger fragments of DNA here. And then as we get towards this last tube, it looks like we're recovering more and more of the smaller fragments that are in the sample. If you look through what happens with the flow through. So this is the flow through that we put through the second silica column, and this flow through went through the third silica column, and so on and so forth. This row here is represented here. So this is the initial extraction where we recovered, where our recovered concentration is 31.4 nanograms per microliter. In this first tube here, with the flow through, we recover 15, almost 16 nanograms per microliter of DNA. And then we're still recovering quite a bit all the way through. There's some pretty low values down here with these last 2 tubes, but we're still detecting DNA even all the way down through this part of the experiment. So these are all of the tubes that we use in the study. And this is all of the qubit values. So what you can see here is this one here. This is from the first spin column. This is the first elution from the second spin column. This is the third, fourth, fifth. So each of these peaks are the first time using a new spin column and then what you see after. And you can see where qubit actually detects DNA, but there's some very small dots. You can see there's a little bitty blue dot there for small concentrations. And then when I plot out the smears from the fragment analyzer, you notice that more is detected from the fragment analyzer. More of these tubes that look like they were empty, there's small amounts of DNA in them as well. But with this large, we can see that the larger fragments are taken the earliest off of these silica columns, and then we start to get smaller fragments further on down the line. But there's DNA in quite a lot of the sample, which you kind of expect because we're sort of overloading the tube, but we wanted to see where the DNA actually traveled when that happened. So I'm just going to explain this chart real quick because we're going to see this later. And this is representative of the amount of DNA recovered. In this case, we did recover, if you calculate the nanograms of DNA, we should recover. All of that is recovered through the 100 tubes that we examined. Most of that is recovered off of the first set of first silica column, about 56%. But then we still recover quite a bit from the later silica columns. So all the orange ones are representative of what we get off of the second silica column, third silica column, fourth, and so on. If you look at the standard of a high concentration that's degraded, so our sort of experimental state, you can see pretty quickly that detecting DNA in these samples is much more difficult, especially with the qubit, despite the fact that this is at a high concentration going in. So this is only about 8 nanograms per microliter here. We see DNA present in more with the fragment analyzer, but it's still not great. And what you end up with this standard, we only recover about 19.5% of the DNA. So that's kind of interesting. Our experimental state that you would never see in any real world setting performs the poorest on this. And only about 11% or 11.4% of recovered DNA. Remember, this is all of the DNA that's recovered only. That first silica column, you can only get 11% of the DNA off of, even after moving it through all of the 10 tubes. When you're looking at standard 2, which is representative of, say, a touch sample of a fresh touch sample, so something that you would expect large fragment lengths still being present, but at a low concentration. There was only 1 or 2 of the tubes that actually had detectable amounts of DNA using qubits. So here you're just looking at what the fragment analyzer picked up. Overall, we recovered about 75% of the DNA in these 5 tubes here that you could detect DNA in. This here is the standard going in. And most of that was from the first silica column being moved through 3 different collection tubes. Oh, I think, nope, that's right. Oh, sorry. When you look at the last standard, standard 4, we recover a little bit less DNA. So standard 4 is representative of our degraded and low concentration DNA sample. We recover about 50% of the DNA in these 5 tubes. And again, most of the DNA was recovered in this first silica column, moving it through 3 different collection tubes. And DNA wasn't detected in any of the other tubes. So 5 out of our 25 tubes for both of these low concentration samples actually had detectable amounts of DNA. So as a summary, what we see is that DNeasy performs better with intact DNA at high concentrations. So our standard 1 performed really well. We recovered 100% of that DNA by using multiple tubes, right? The larger fragments tend to be recovered at a higher frequency in the early stages of the experiment, and then we start to retain some of the smaller fragments. And it does a really poor job with this experimental state 3Z, where we have both high concentration and highly fragmented DNA. It actually did better with what we would think of as degraded or low copy number standard, but by far it did better with the fresh DNA, which is which is what we usually use it for. So I guess that's probably a good thing. So I can't read my screen. There we go. So what we see is that DNA is lost during multiple stages of a DNA extraction. So it's obvious that the DNA doesn't necessarily bind efficiently to the silica column. Maybe the fragments are too small, maybe it's crowded in some places, but not all of the DNA can bind efficiently to the silica column, or it's not sufficiently removed from the silica column during the elution stage. Oops. We also know that DNA recovery is dependent on sample type. I'm really excited when we can actually get some of this stuff out there, because I know with all the graphs that we were looking at before of all the different extraction methods, really what we're seeing is that, one extraction method works really well for one type of sample, whereas it doesn't work so well for another type of sample. So that's important to note, especially when you're thinking about using precious samples in the future. And unfortunately, our second part of our goal, when we try to modify the protocol by either putting the eluate back through the silica column to maybe increase the yield, or putting the flow through back through, it didn't actually increase our DNA yield like we had hoped. So we're still working on tinkering with the modification for this particular kit. And I am going to pass this on to Dr. Kemp so he can give some closing remarks. Oops. I think you're muted, Brian.
Yep, Dr. Kemp, you might be muted still. Let's see if I can unmute you from here.
It's funny how that works.
There you go.
Yeah. Some messages we would like to relay to all the audience, some take-home messages are this. One, I hope that we've illustrated that the issue of DNA loss is certainly underappreciated and poorly understood, even after our experiments. I mean, we're still learning about this, but it's far from resolved. Second, as reiterated numerous times, I think, through this presentation, is that there's considerable variance in DNA loss and fragmentation that can occur during DNA extraction and purification. And as Dr. Beatty said, it would be hopefully after the publication of our results, and maybe as others follow our lead, that we could have some information going into a project knowing I have this type of sample and this type of extraction method works best, rather than just pulling something directly off the shelf, but knowing something that it's going to do to the quality and the quantity of the DNA that results, especially for highly valuable samples. In the case of silica-based extractions, as Dr. Beatty illustrated, DNA loss occurs both because of the efficient binding to silica and failure to efficiently remove the silica, to remove the DNA from the silica once it's bound. I believe she failed to mention that we have done a couple of experiments to try to recover more DNA off of columns, but nothing seems to be working. We can identify where the DNA is. We're just having a hard time recovering it into a single tube. We can get it across the 100 tubes and find 100% of the DNA, as we saw in standard 1. But how do you get that into a single tube without having to combine those volumes, which would dilute out the sample, which is not the point. And then lastly, we advanced the notion of approaching these issues using a standard methodology. And we're pointing here to commercial labs and academic labs. So the idea of using a known quantity and quality of a DNA sample to put into a system, an extraction system or whatever, and seeing what comes out is highly desirable over what has been more traditionally done, where extraction 1, 2, and 3 are compared to each other, and we find a relative best method that doesn't tell us about the absolute efficiency. So we can improve upon that using a standard methodology approach. I think I speak for Dr. Beatty and Dr. Monroe. Thank you so much for your attention, and we hope that everybody is doing really, really well out there. We know, we're having a hard time with COVID and working in the lab here, and I'm sure you guys are experiencing the same. And we just hope that everybody is staying safe and staying healthy. And lastly, I would like to acknowledge the National Institute of Justice for the support for this research. You can see here the award that was given to myself and Dr. Monroe, and their disclaimer. Anyways, that's it. Thank you.
All right, thank you so much, Dr. Kemp and Dr. Monroe and Dr. Beatty. We will go ahead and start our Q&A, but before we do, I do want to make sure everyone is aware that we are recording today's event, and we will be turning that around into an archival version over the next few days. I'll go ahead and take back over control of the screen. So we will be converting today's live event into an archival so that you'll be able to have access to that and come back and either refresh on the content or provide to any colleagues that might have missed today's live event. Also, we do not have the slide deck available, but all of our presenters were gracious enough to give us some additional resources that we are going to disseminate out to you, our audience. So make sure before you leave that you do go back to the main event page that you came into today's webinar, and you'll be able to download those directly after the event. So Dr. Kemp, the first question came in at the beginning while you were presenting. Yeah. They were asking if there is any explanation on why Lee et al. Study showed more extraction from degraded standard than intact.
Yeah, so I quickly looked into that paper while we were presenting and wanted to just quote the paper. I'm just going to tell you what they say. It doesn't make particular sense to me, but I'm just going to read it anyways. So they say, on the other hand, as buffer PB in the QIAQuick PCR purification kit is optimized for efficient binding of PCR products as small as 100 base pairs, its application to the QIAamp mini spin column was expected to allow efficient recovery of degraded DNA, and the results confirm that observation, but that doesn't really tell us why. So, but that's what they said.
Thank you so much, Dr. Kemp. That is extremely impressive multitasking to be able to go and find that while we're presenting. So I definitely appreciate that extra effort for our audience. Dr. Monroe, the next question came in while you were presenting, asking, did you test phenylchloroform method in your study against the 4 DNA standards?
No, we didn't, but actually that, since it's being used so much in forensic laboratories, I definitely think we're going to include that into the set that we've already done, as well as including other viral columns, since the viral columns with Rowland et al. Seem to work pretty well across all standards, as well as the PrepFiler bones, tissue, and adhesive. So it's slightly different, but still has that bead-based extraction method. So I think we're definitely going to expand upon the methods that we've already done.
All right, the next question again came to you, Dr. Monroe, asking if you could explain or justify why ancient methods were superior over the other 2 in all of your samples, whether fully intact genomic DNA or degraded genomic DNA.
The 2 best performing, so I should clarify that lots of the ancient methods didn't perform all that well. So take that with a grain of salt. I think the 2 best were the 2 Rowland et al. 2008 methods. that were using viral columns. And I find that very interesting because some other studies, and the names are escaping me right now, but had found high yields using silica-based columns that were specific for viral extractions. And so there might be something with those types of kits that make the yield so much better. The buffers, I don't think, make that much of a difference because the Roland et al., one of the buffers they use is the same buffer they use in Dabney et al. And Dabney basically is mimicking a buffer from Qiagen for the Miniloop kit. So in some cases, they're using the kind of same reagents. And so maybe ultimately what makes something better or worse might be something very specific. So in that case, they take the column from that kit, don't use the rest of it, and then use buffers from other methods. So I have a sneaky suspicion in that case it performed well because that silica column for viral extractions is really good. And then I think, in other cases, I think in this case it was luck because they were very clear that their goal was to look for short fragment sizes, not larger ones. And I think it was just an added benefit that it did work across all standards.
All right, perfect. So this next question came in at the end of Dr. Beatty's presentation, right around the beginning when Dr. Kemp came back on. So really it can be posed to either of you, whoever wants to take this one. Do you combine the elutes to increase concentrations?
Not for the part of the project that I talked about, no. The idea was to see if we wanted to keep the volumes consistent across all the tubes and see how much DNA was retained in each of them to keep it consistent. And then we back calculated nanograms in each of those tubes to figure out how much was retained in them. But when we try to modify our experiment, using the modifications on the protocol, we did things like try to use less buffer in two different elution steps to try to end up with the same amount of volume but with increased yield and things like that, and it didn't really work. It wasn't working well enough to make it worth that extra step, if you know what I mean. So yeah.
But to reiterate here that, let's say with standard 1 in the experiments that Dr. Beatty talked about, I can't remember the exact number, but let's say that there was detectable DNA in 25 of the 100. If we combine those 25 at 200 microliters each, we're going to just end up with a huge volume of super dilute DNA. And so that defeats the purpose, right?
Right.
All right, next question is asking, could hypochlorite solution cause DNA loss in bone sample?
Well, we've published, I've published 2 papers on the influence of decontaminating bones with sodium hypochlorite. We're talking about, I think we're talking about bleach here. What we found was it was very, for methodological reasons, you probably have to read the paper, but it was very difficult to determine the amount of degradation that the bleach was doing to the DNA inside the bone. It's very, very hard to estimate. So as far as I'm concerned, and at least my knowledge of the literature, I don't think that it's been demonstrated, although it's talked about a lot that bleach would do damage, but demonstrating it is a different thing. And I don't think it's been demonstrated.
All right. Next question, again, is back to you, Dr. Kemp. They were actually back-to-back questions on the same topic, asking if you could comment something about the DNA collected from the touch documents and how the touch DNA was simulated.
Nothing was touched. One of our standards in our sort of in our conceptualizing these different standards. One, we just wanted it to be low concentration but highly intact, so that, like, I'm in my office right now. If I touch my desk, I leave highly intact DNA, but it's in low copy number. So we actually didn't test touch DNA. We were trying to simulate it with one of our standards.
And our standards were at a slightly, they were at a higher concentration than what you would expect with touch DNA, but that was so that we could detect it using the methods that we were using.
Right. So we had to deal with the fact that the fragment analyzer has a certain sensitivity, and 1 nanogram, if we lost 95% of it, we wouldn't be able to see it on the fragment analyzer. So yeah, Dr. Beatty's right. We ended up using standards that maybe others would not consider particularly low, but they were lower.
All right. So we had quite a few comments once you finished up about how informative your presentation was. Excellent, awesome presentation. So obviously we want to make sure that all of you as presenters know that.
Thank you. That's so nice. Thank you.
Thanks, everybody.
Next question is going out to the group. Qubit versus nanodrop. Which is better for postmortem samples, in your opinion?
I'm gonna have
that was my sigh. I've used both, but I haven't. Go ahead, Kara.
I would say that if you have any samples with impurities, you're better off with the qubit. So I don't know what postmortem samples you have, but nanodrop is just so sensitive to any sort of impurities that you have. It's very finicky. I would say, even though we're still having issues really looking at best ways of quantifying things, I would say qubit is probably better. Now, if you have to have high throughput, then maybe the nanodropper you can maybe do time or something like that. That might be a thing you might have to consider. But I would say just as far as consistency and its ability to resist some inhibitors, I would go with the qubit.
All right, perfect. With that, we are going to go ahead and close things up for today. Obviously, the first thing we would like to do is to thank our subject matter experts, Dr. Kemp, Dr. Monroe, and Dr. Beatty for their presentation, as well as all the research that they've put into their NIJ grant. So thank you all three very much for being here today and taking the time out of your day to present.
Thanks.
Yeah, thanks for having us.
All right. With that, we would also like to take this time to thank our attendees for joining us today. These webinars are simply not possible without your attendance, your support, and your interaction in our Q&A. So thank you everyone for joining us and asking such great questions for our subject matter experts. Before you leave today's webinar, we always ask that you participate in our short survey. You'll see the word survey directly below the closed captions pod on the right-hand side. Simply click the word survey. It will open up a new window for you, and you can give your feedback on today's event. That feedback is always vital information that we use to make sure we are providing you the best webinar experience possible, as well as adjust the content and the way that we disseminate out that information to you, our audience. So if you could take a few moments to complete that, we would definitely appreciate it. Also in the chat pod, you will see some links coming through from Tom, our ghost host, putting information in there to follow us on our social media outlets, Facebook, LinkedIn, and Twitter, as well as our website where we're constantly adding new content such as webinars, news reports, and success stories. Also, we have our podcast on our website as well. If you're not following those podcasts, make sure that you are. We had over 40,000 listens on our podcast so far this year, and we are still growing that continuously. So make sure you're following us so that you can stay up to date on all the constant new material that we're putting out through the FTCOE to the forensic community. Again, we have recorded today's event. We will be converting it into our archival, and also make sure that you check out back on the main page the additional resources that our presenters were gracious enough to provide today as well. Again, thank you everyone for joining us today. We hope you found this information extremely useful, and we look forward to seeing you in our next webinar. We hope you have a great rest of your day.